Hello friends, welcome back to my channel, and today in this video we are going to study about hematopoietin class 1 family cytokines. So in the last video we learned about interleukin 1 family of cytokine. Today this is the second family, that is hematopoietin class 1 family cytokines. Now if you see the general feature of this class, then these are small soluble cytokines and they helps to communicate between and among cells of immune system. And this hematopoietin cytokine family, it is also known as class 1 cytokine family. Now the cytokines of the hematopoietin family: interleukin 2, it helps in T and B cell proliferation. Interleukin 6, it helps in B cell differentiation into plasma cells. What are plasma cells? These are B cells that secretes antibody. Interleukin 4 versus interleukin 2, it helps in the differentiation of T cells along one versus another differentiation pathway. GM-CSF and G-CSF, they helps in the differentiation of particular leukocyte lineage. Cytokines such as interleukin 2, 4 and 3, they have short helices of 8 to 10 residues in length. 6 and 12, they have helical lengths of 10 to 20 residues. So this is the individual function and little bit of structural details of hematopoietin class 1 family cytokines. Now coming to the receptors, they are divided into three receptor subunit, that is gamma, beta and GP130. Under gamma family, interleukin 2, 4, 7, 9, 15 and 12 recognize these receptors. For beta it is 3, 5 GM-CSF. For GP130 it's interleukin 6, 11, LIF, OSM, CNTF, interleukin 27. So first let's see the gamma bearing receptors. So as you can see in the diagram, if only alpha segment is present, it has low affinity for interleukin 2. If alpha, beta and gamma, all the three are together present, then only it is a complete receptor and it has high affinity with the interleukin 2. If gamma and beta present, then it will have the intermediate affinity. So with only alpha it is low affinity, with gamma and beta it is intermediate affinity, but with alpha, beta and gamma it shows high affinity. Now coming to the subunit composition, here it is beta and gamma, here it is alpha, beta, gamma, this is only alpha. Dissociation constant, if you see, 10 to the power minus 9 here, intermediate 10 to the power minus 11, high 10 to the power minus 8, the low. The cells that expressed for this, it is NK cell and the resting T cells. These two are expressed by activated CD4 plus and CD8 plus T cells. That means we are talking about TH and TC cells, and also in activated B cells. Now here it is written that the high affinity and the low affinity both are present in activated cells. Now, high affinity, we can understand that these are present on the cell so that it can bind to this interleukin and respond accordingly. That is proliferation. Whenever any antigens entered, it needs proliferation, it needs differentiation, maturation. So it is understandable why the high affinity receptors are being expressed. Now, in these activated cell, you have to tell me in the comment section that why the low affinity receptors are present. What is the function of the presence of low affinity receptors on these activated cells? This you have to tell me in the comment section. About these low affinities or the inhibitors, I told you in the last video only. So just either search the literature or use your knowledge or see my last video to know why these low affinity receptors or any inhibitors are also expressed on these activated cells. Now one disease, that is X-linked severe combined immunodeficiency, it occurs due to defect in gamma chain gene. Now coming to the next one, that is this beta subunit receptor family. So here the interleukins are 3, 5 and GM-CSF. So in the alpha, if only alpha is present on the surface, then it will show the low affinity with the interleukins. If beta is associated with the alpha, then now this is the complete receptor molecule showing high affinity for the interleukin. Now whenever interleukin 3 or GM-CMS will present, they will bind if alpha beta is present. So first they will come and bind near the alpha. Once they bound with the alpha, then this rearrangement of beta part of this complete receptor will occur, and now this alpha beta together they will form the complete receptor with high binding affinity with the respective interleukins. So first the alphas are aggregated on one side and the beta parts are aggregated on another side. Once these interleukins are present in the close vicinity with the alpha part, it binds with the alpha part, and this beta part will rearrange, shift, and come near to the alpha part. And that's how it gets activated. Now, interleukin 3 and GM-CSF both act on hematopoietic stem cell and progenitor cells. They activate monocytes, induce megakaryocyte differentiation, and all three of these cytokines induce eosinophil proliferation and basophil degranulation with release of histamines. So, this is the function of interleukin 3 and GM-CSF. Coming to GP130 receptor family, it has two, interleukin 6 and interleukin 12. Interleukin 6 helps in the initiation and 12 helps in the differentiation of T helper cells. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.